This is a summary of a paper of mine from a few years ago entitled Serotonin Signaling in the Gut and Autism Spectrum Disorder, a Translational Mathematical Model. This is yet another one of those projects that started with me finding a really cool experiment that I wanted to describe, and I ended up going down a rabbit hole. I'm going to get to the experiment soon enough, but first I wanted to give a brief history of autism. Okay. So the term autism itself comes from the Greek word autos, meaning self, and it was actually first used by psychiatrist Eugen Bleuler in 1908 to describe a schizophrenic patient who had withdrawn into his own world. Next, it was described by Leo Kanner in 1943, and then also around the same time by Hans Asperger, 1944. And of course, we're all familiar with the term Asperger syndrome. Leo Kanner studied 11 children who had difficulties in social interactions, to adapting to routines. They had sensitivity to stimuli, especially sound. They had food sensitivities. A lot of these kids had good intellectual potential, and they also had echolalia, so repeating words and phrases that someone else says. Asperger worked independently, and he studied a group of children who resembled these descriptions but did not have echolalia as a linguistic problem, and they also spoke like grown-ups. Then in 1952, Diagnostic and Statistical Manual of Mental Disorders, the DSM, kind of the Bible of mental health diagnoses and disorders. This is when the term autism was introduced, in 1952. Although kids with autism actually continued to be diagnosed under various terms and conditions related to schizophrenia in the DSM and also in the International Classification of Diseases until the 1970s. And even if you talk to anyone, everybody knows that until relatively recently, I guess until about 30, 40 years ago, a lot of times kids with autism actually were misdiagnosed as having schizophrenia. By early 1970s, it had become widely recognized that autism and schizophrenia are actually distinct mental disorders. And this was formalized in DSM-3 in 1980s for the first time. This is when we got a new diagnostic category. Asperger's syndrome was introduced in the DSM as a formal diagnosis in 1994, so this is actually relatively recent. In 2013, Asperger's syndrome and infantile autism were then unified into a single diagnostic category, ASD, Autism Spectrum Disorder. That is what we use right now. And it really provides a single name and a set of criteria for conditions that were previously defined separately, such as Asperger disorder, autistic disorder, pervasive developmental disorder that is not otherwise specified, very strange name, nevertheless childhood disintegrative disorder, Rett's disorder. So a lot of these things until 2013 were classified as separate things, and now they are classified under the single umbrella of autism spectrum disorder. One of the possible explanations for why we now are seeing what appears to be a rise in ASD diagnoses, they were classified as different things before. Okay, so now to the experiment that actually got me really interested, and to the biology that got me really interested in this topic about gut, brain, and serotonin. I was very interested to learn that serotonin is actually one of the first biochemical markers that was linked to autism. And in fact, hyperserotonemia, so really high blood serotonin levels, has been found in like a quarter of individuals with ASD. And it was one of the most consistent peripheral findings across studies. Again, just a quarter to a third, but still, this is quite non-negligible. Paradoxically, this elevation doesn't directly reflect brain serotonin levels, and I'm going to get to that momentarily, because serotonin is produced in two main places. Only about 5% of it is actually produced in the brain, but 95% of serotonin is produced outside of the brain, and mainly in the GI tract and by the with the enzyme TPH1. So that was kind of interesting. Somebody was describing it that this is where we get most of the serotonin, but this is the serotonin that makes a life worth living. An interesting thing about the blood, about the brain serotonin is it does not cross the blood-brain barrier. So whatever we measure in the blood is actually what is produced in the gut. So central and peripheral pools are actually separate. Interesting. How do we measure it? Well, serotonin levels in the brain cannot be measured in the blood because of a blood-brain barrier, because these are separate tools. And in the periphery, serotonin levels are actually measured in platelets, where it is highly concentrated. But platelets do not synthesize serotonin. They actually pick it up and absorb it using the SERT receptor. It's going to be very important. We're going to come back to it a lot. And this uptake occurs as platelets circulate through the gut. So once inside the platelet, serotonin is stored in these dense granules and is released during activation, like when platelets get activated, for example, for the purpose of clotting. So this is kind of a very basic diagram. So SERT receptors in the blood, and the platelets would have some of these receptors, and they would pick up the serotonin that is produced in the gut. There are SERT receptors also in the gut for its own things, so it is expressed on multiple cells. Oops. Serotonin can act as a growth factor in the enteric nervous system. It is also known as the second brain, and this was really not popularized, but really put together very extensively in the work by Michael Gershon. He even has a book called The Second Brain about the enteric or gut nervous system. If SERT receptors are hyperactive, they take up too much serotonin. And then, so if they are hyperactive, for example, in platelets, then a lot of serotonin gets picked up by the platelets. So you have elevated serotonin in the blood and reduced serotonin in the gut. Just different pools, you get distribution there. And this can affect the enteric nervous system because platelets have taken up a lot of the serotonin there. So it's not available for whatever its purpose is in the gut to actually help act as a growth factor in the gut for the enteric nervous system. Yes, this is the book, The Second Brain by Michael Gershon, that I was referring to before. And so this kind of redistribution of serotonin from different parts of the body, specifically from the gut to the platelets, that can lead to some interesting outcomes. And here we come to this experiment that was so interesting to me. The Margolis, as I call it, mouse model, because the first author is, that's the last name of the first author of that paper. That's a paper from 2016. So, as I said before, SERT is a transporter that clears serotonin from the extracellular space, which is expressed both on the gut and the platelets. So SERT is expressed in multiple places. And then there is this IL-56 SERT variant, and it is a mutation that increases serotonin uptake. And that can lead to hyperserotonemia, or high blood serotonin, because, once again, platelets keep taking up a lot of the, if it's overexpressed, they're sucking out all the serotonin, or not all, but a lot of it from the gut. Therefore, you see a lot of serotonin in the blood and not enough remaining in the enteric nervous system in the gut to actually help grow the enteric nervous system. And this was the mouse experiment that was done by Margolis and colleagues in 2016. This is a publication over here in case you want to look it up. So the authors compared 3 groups of mice. One group were the mice that were engineered to express this IL-56 SERT overexpression. So they were called also SERT positive mice. So these take up a lot of serotonin, so much that there is not enough left in the gut. There are, of course, wild-type mice, then what we call normal SERT, and knockout mice, so SERT negative. And this was my little diagram for them. What did they observe? Well, the SERT positive mice, they had elevated blood serotonin, which of course makes sense because it was taken from the gut and transferred via platelets to the blood, to be measured in the bloodstream. As a result, they had less serotonin in the gut, and since it acts as a growth factor in the enteric nervous system, their enteric nervous system was reduced. According to the data that they had, it was about 60% of wild type. They had slower GI motility, they had altered reflexes, and this was really interesting, that they had ASD-like behaviors, like repetitive actions, impaired social interactions, things like that. The SERT knockout mice, well, they had lower serotonin clearance, obviously, and as a result, there was more serotonin circulating in their gut, so they had expanded enteric nervous system. In this particular case, it was about 120% of the wild type. Again, those are proportions that I was able to get out from the data in the experiment. And behaviorally, there were no ASD-like behaviors. Although some other studies have noticed increased maybe anxiety-like behavior in these knockout mice, but not the classic ASD phenotypes. And if we want to kind of put together a little summary of this, so the knockouts, they had larger enteric nervous system size because they had too much serotonin floating around, so less serotonin in the blood, more of it in the gut. GI function, okay, so motility was a little higher, at least based on past data. No ASD-like behaviors in this study. Wild type we take as baseline, SERT positive, which means that platelets are taking up a lot of that serotonin, taking it back in the blood, less remaining in the gut, so less of a growth factor for enteric nervous system. Motility reflex and GI were worse, so they had GI issues, and they had some ASD-like behaviors, lower social behaviors and higher repetitive actions. Okay, interesting. So then the authors wanted to look at proof of mechanism. So is it really the serotonin situation? And then if it is, can we modulate it? So one of the things that they tried was this fluoxetine, which is an SSRI, and it's a SERT antagonist, so it blocks some of the SERT receptor. And by giving that, you can mimic some loss of function in SERT activity. So what they did is they administered it to pregnant mice, and as a result, they saw ENS hyperplasia in pups because they were given to the mice. So animals became similar in this way to knockout mice because you blocked out the activity of the SERT receptor. Makes sense. So in this case, this was a confirmation that reduced SERT would lead to increased serotonin signaling. Again, makes sense. Now that you have serotonin floating around, you would have enhanced ENS development. Okay, so just a little summary. So you took wild-type mice and you blocked their SERTs, and so they started acting like knockout mice. Interesting. The reverse experiment here was, what if can you compensate for under-signaling in the gut in these SERT positive mice, so these alpha-56 mice. So they administered to these SERT positive pups, they administered this serotonin receptor agonist. So antagonist would block the activity and agonist would try to amp it up and increase it. And what we saw is they rescued ENS hypoplasia. So yes, a lot of the platelets that were taking up the excess serotonin, they were still taking it up, but they were trying to boost the activity of the SERT receptors in the gut to kind of compensate for it, and they were able to rescue some of the hypoplasia. And what is even more interesting, that improved some of the social behaviors in this SERT positive mice. It's partial behavior rescue, but still. So to summarize, you had SERT positive mice, so they had smaller enteric nervous system, these pups, and they had ASD-like behavior. They were given this serotonin receptor agonist, and the goal was to compensate for under-signaling. And they rescued the ENS hypoplasia, so basically allowed for the enteric nervous system to get the growth factors, get the growth signaling, and that is what is really interesting here. It seems to have improved social behaviors, brought them closer to the norm. Did not fully rescue them, but it did help. Interesting. And the key message of kind of these experiments is that serotonin signaling can affect the development of the enteric nervous system, and it can be modulated through multiple pathways. Very interesting. Now, can we quantify this? Well, since I'm a modeler, all I want to do really is just describe things with equations. And so this is what I worked on here. I wanted to develop a mathematical model to capture how serotonin signaling over time can influence the growth of the enteric nervous system. And I have this pretty simple model. So what do we have here? I'm just modeling the gut serotonin, and I have some inflow term, some synthesis, and I have some clearance term. And I have this term here, which I called my SERT term. This will be my parameter of SERT, I guess, expression. And so as much as, and this term is going to affect, so this is the clearance of serotonin from the gut. Okay, So this is what will go into the platelets. Whatever is remaining is what's going to actually go into contributing to building the enteric nervous system, quote unquote, size. So however that is quantified. And it will be increased proportionally to gut serotonin and obviously to the current ENS size. So if I have a lot of this SG remaining, then I will have more building blocks to work with to build my enteric nervous system. If a lot of this gets cleared out and then taken out in the platelets, obviously I will have less to go on here. And the main question is, what will affect the size of the enteric nervous system? You do not need to be a modeler or a genius. You can just do some thought experiments. So before I actually run any simulations, I like to think through what it is that I expect. So as I said, if SERT is high, then we will get higher serotonin clearance, so less serotonin available to contribute to a formation of enteric nervous system, and therefore smaller ENS. Okay, but if I have a lot of the synthesis, yes, I have high clearance, but if I have high synthesis, then maybe it will balance out. Or if I have this parameter of increase of the enteric nervous system proportional to serotonin that we have, well, if this is large, then maybe we can do a lot with even limited serotonin that we have floating around. If SERT is a wild type, but for example S0 or these guys are low, then we will get smaller ENS. So yes, maybe this is still whatever is called the normal expression, but if these things are not compensating or balancing them out, then we might still get smaller enteric nervous system. Or as I said, if this one is really small, but these are kind of really large, so maybe we get some large enteric nervous system because maybe we can compensate with smaller production term or smaller KN, etc. So there are these interplays. It's not just about the expression. It is really how these things are interplaying with each other and how they will balance each other out. Okay, so now I want to parameterize my model as much as I can. I was primarily working with a Margolis experiment. The authors there examined mice at about 6 to 8 weeks after they were born, and then they recorded the relative differences in the enteric nervous system. This is an example from one of the figures where they were comparing wild-type and SERT-positive mice, and very roughly speaking, these are your SERT-positive ones, the ALA56 ones. They're about 60% relative to wild-type. So this is where I was getting those numbers. So that's where I was getting this. And same thing with the knockout mice. Again, figures like this, that's pretty much what I had available to work with. So when you looked at wild type versus knockout, they were about 120% of the norm. Okay, very minimal data, but we do what we can. So then I parameterized my model to capture the following day 50. I said mice were examined at 6 to 8 weeks. 50 days is approximately 7 weeks, kind of in the middle. And I was playing with different parameters to really capture this main behavior, that I would have my wild type would be about 100% by 50 days. And then my SERT plus would be about 60% around 50 days, and then my SERT knockouts would be around 120% at the time when this was measured. So this was really the benchmark and the calibration of things that I was working to try to benchmark the model simulations. I do not have much more to work with, but still, this allows me to capture the key observations from this original data, which is that variation in the SERT-mediated serotonin clearance can affect the size of enteric nervous system at the reported time. So it's basically playing with the value of my SERT parameter in order to calibrate the model to this. Okay. Now I described mice. That's nice. How can this actually map to people? Not that this is something can be used clinically, but I still would like to try to get a sense of these things. And translating findings from mice to humans, especially for developmental things, requires aligning of developmental stages, not just linear time frame. Mice live about 2-3 years. Humans live 80 years around so. But you can just map it one-to-one because it's not linear. And here, for example, ASD is typically diagnosed before age 5, so modeling early developmental dynamics is crucial. What I ended up doing is I actually found this great paper by Dutta and colleagues. Here's the reference, men and mice relating their ages. And the authors here, they, instead of using uniform time conversion, they actually aligned mouse and human lifespans by developmental milestones. I reworked the data from that figure, so that while this is my figure, this is completely based on the data and analysis done by the Dutta paper. I'm trying to show here, so okay, like how long the gestation would be mapping wise. Birth, we're starting at 0, weaning, puberty, adulthood, middle age, etc. And we know that so experimental timing was about 6 to 8 weeks, and that will actually map to approximately, according to this, actually about 11 to 13 human years. So if one were to use this to design an experiment, maybe they should have looked at maybe a little earlier, closer to what would be 5 years, so maybe that would be better. But it is unclear from the paper, from the Margolis paper, if they saw any ASD-like behaviors by 5 weeks or not. Maybe they did, maybe they just wanted to observe them at this, just make sure that everything that could have developed has developed. I don't know, but this is what we have to work with. And then if this enteric nervous system size is indeed linked to this type of ASD, because again, this is not just ASD, it's a spectrum. There are a lot of different manifestations. This is a subtype, but if ENS size is indeed linked to this type of ASD, then we should see these differences by age 5. So the human model, if I were to try to make this a little bit more translational, would have a lower enteric nervous system size for my simulated kids with ASD by age 5 compared to non-ASD. Okay, so then I was lucky enough to find this other experiment from Giugani et al. From 1999. This is the reference. Please feel free to look it up. And this person, this team, they reported key differences in serotonin synthesis capacity between kids with and without ASD. And they were showing that in kids without ASD, serotonin synthesis actually peaks by 5 years old and then declines. So these were the data, and then I wrote a kind of a little functional form, so a model to try to capture the basic trends. In kids with ASD, serotonin synthesis actually seems to increase more gradually and peaks later, and it does not decline as much. So it, I guess, stays up. So in order to capture this in the model, I replaced the synthesis term as 0 that I had before with this time-dependent serotonin production term, which I am describing here. So this is non-ASD, this is ASD. The data were in serotonin synthesis capacity, so-called K-complex over time. And then if you kind of overlay these, this is what they would look like. And you can see, okay, non-ASD, they have higher peaks, but then goes down. And in ASD, it actually lower peak, but then it stays up more. These are not perfect fits, but this was sufficient for my purposes. I finally get to show my model analysis, because my main question was, what is the impact of varying model components, varying different model components in individuals on the size of the enteric nervous system? As I said, so I modified the model to replace my constant serotonin production term as 0, which was just a constant, to this dynamic one that I wrote and parameterized for simulated kids with ASD and non-ASD. I do not know about how other parameters of features scale and translate. So for now, I'm assuming that all the other relationships are largely preserved. This is a big assumption. If there is more information on it, this can be adapted, but this is what we're working with right now. And then parameters of interest, so just recall this is my system here, is this Kn, which is the serotonin-mediated expansion of the enteric nervous system, and my SERT parameter right here, which is the clearance, essentially, SERT-mediated serotonin uptake from the gut, so how much of it is cleared, leaving, and whatever is left is what's going to go into the formation of ENS size. And I'm not giving the functional form here because it's not very attractive of what S0 is now replaced with for ASD and non-ASD kids. It is in the paper, you can look at it. But basically, A would be parameter A, there is a rate of serotonin synthesis in the human model. So it's kind of an updated function that replaced the 0, and this was the key parameters for that. And the approach that I did here is what we call a parameter sweep. So I vary parameters along some range, and then I record my simulated projected ENS size at 5 years. Okay, so let's look at, this is kind of the main result here. So on this axis, we have the serotonin synthesis parameter. It's right here. And on here is my Kn, so this guy over here. And on the z-axis, we have the cert. And then the points that I'm recording is that percentage of ENS size of the norm, percent of the norm. Okay, so what do we get? And then the surface that I put here, I actually ended up doing that recently as I was preparing this talk. So the surface didn't make it into the paper, but I was using the same code. And so this one is the 100% ENS. So this is kind of the norm. And the really main thing that I was kind of looking at and getting here is you can get 100% enteric nervous system, so what would be what we take as the norm here, with multiple parameter combinations. Again, we kind of could predict that with the thought experiment we talked through, but it's still helpful to actually see this. And I don't know if this is a better visual because there are a lot of dots, but to see that this is the surface, it's not a, aha, you got to be here and then that's that. A lot of these components will be compensating for each other or interplaying. So it's not just you have height cert clearance, therefore you're screwed. No, because maybe other things compensate or other things play out. And these are just 3 of the components. We're going to get to scaling of these things in a moment. But before we talk about that, the next natural question would be, well, okay, so we have this observation in mice that small ENS, small enteric nervous system, is somehow affecting the brain, and that is correlated with actual issues with like cognitive processing. How? Well, the mechanistic link, just physically, seems to be the vagus nerve, and it seems to be the main gut-brain connection. So this is this is a figure from a recent paper that came out. There's basically this big connection between the gut and the brain. And it's a bidirectional neural conduit between enteric nervous system and central nervous system. An interesting thing, 90% of vagus nerve fibers are efferent, so they are carrying sensory and chemical signals from the gut to the brain. Only 10% actually sent from the brain to the gut, but most of the information, most of the signaling is going up. Interesting, because these gut-to-brain signals, they can apparently influence neuroinflammation, appetite, energy metabolism, mood and behavior, a lot of things that are being influenced by this direction, by the signaling that comes from the gut nervous system. And the ENS neurons are necessary for generating these signals. So it generates this neural activity and molecular signals that will travel up here. It also interacts with the gut microbiota and with the immune system and the endocrine pathways. All these signals are shaped traveling upstream. So, and again, we all, or not all, but if we follow the literature, we see that, interesting, it seems to be gut microbiota differences between in kids with ASD versus not ASD. And a lot of other these things, well, maybe this is all part of kind of the same story of enteric nervous system and the connection between the ENS and how it travels to the brain and how this would actually affect what happens in the brain. And then within this paradigm, a hypoplastic enteric nervous system, so a small one, might translate into reduced signaling bandwidth, because I'm assuming that ENS hypoplasia would mean fewer enteric neurons, so less input into the efferent vagus nerve, so the ones that are traveling up to then signal all the things like neuroinflammation, appetite, and energy, and mood and behavior. And maybe this will lead to dampened, unaltered signaling in the brain. And of course, this is especially relevant during early neurodevelopment, when brain circuits rely on external input for tuning and maturation. Interesting. So, continuing on this, though, what would be the consequences for CNS development in a world where this is a mechanism that can influence the brain? Well, some of the consequences for CNS development could be issues with metabolic regulation, inflammatory homeostasis. If ENase-based signals are weakened, well, maybe brain will receive inadequate input for regulating metabolism. Maybe mitochondrial function in the brain may suffer. And this happens early on, might interfere with neuronal development and plasticity. Some of my other work, because I went down metabolism rabbit hole, in dementias and Alzheimer's, I am now a supporter of the metabolic hypothesis of Alzheimer's, that the issues with metabolism and specifically with hypometabolism in different regions of the brain can be the initiating event that can start then the cascade of neurodegeneration. Well, maybe if this happens early on, you are getting similar issues, just probably from a different source. Although some of the studies that I saw even suggested that individuals with ASD might be more likely to develop dementia. If this is a mechanistic link, that's possible, but also I do not know how one would actually study that, because we have only been studying individuals with ASD really thoroughly for the last 25 years. The first kind of analysis started in 2000, so I'm not sure where that information would come from really. But one can, in a world where this is true, then one can also expect disruption of neuroimmune, serotonergic, behavioral regulation pathways. And then as a result, this could provide a plausible link to at least, as I said, some ASD phenotypes, where abnormal brain development could emerge not solely from some intrinsic brain activity, but also from impaired gut-brain communication. And maybe this could be also an upstream driver, contributing like reduced vagal tone, altered brain energy balance, disrupted neurotransmitter homeostasis. And I tried to put this together in how could this be happening? Because what we see from the experiment is you might have issues with serotonin synthesis in the gut and clearance and maybe mediated growth. So the things that I was talking about, these are the three components that I just looked at. And in their interconnection, if chips fall not very favorably, you might get some anti-hypoplasia, then some black box happens, and ta-da, you have an ASD phenotype. And the proposed mechanism here is that maybe it is somehow connected to this impaired vagus nerve signaling, which in turn leads to impaired mitochondrial function in the brain, which is general signaling in the brain, and then leading to this, at least some ASD phenotypes. And I guess this is just kind of a summary, as a hypoplastic enase may weaken vagal input into the brain, compromising brain metabolic neuroimmune regulation, possibly contributing to CNS dysfunction. Okay, so in a world where this is the case, what could be some of the approaches that could target this axis? I've seen some interesting information on vagus nerve stimulation, and the idea is that you just physically stimulate the vagus nerve to enhance this gut-brain communication, maybe modulate some neural circuits. These are a couple of references that I found when they were starting to look at that, and at least it looked promising. That being said, okay, this is 2017. This is from 2024, and this was interesting. This had some interesting results, but I have not seen anything dramatic quite yet. But it seems quite promising, and maybe the issue right now is just very localized and not very extensively discussed. This was interesting, the photobiomodulation. Again, so this is another thing that was coming up in my research on Alzheimer's, which on some level could be thought of as the other side of the spectrum of cognitive dysfunction. Again, not to say that ASD is necessarily cognitive dysfunction, but just some of the maybe extreme cases. But this is a thing that has been popping up in my research on both sides here. And the idea here is that this thing applies this near-infrared light to the scalp to stimulate mitochondrial activity and maybe enhance cerebral blood flow and reduce neuroinflammation. And there have been some interesting results here. I also know of a startup that has been doing that. They seem to be, I think it's called Jellica. And they seem to have been showing some very promising results, but something to really keep an eye on. Also, I'm not promoting anything. I am not supported by, big vagus nerve stimulation or something. I'm just sharing what approaches would make sense if one were to try to treat and target this particular mechanism. Again, not saying that this is the mechanism, just that this is maybe a subset of ASD individuals for whom maybe this could be helpful if this is the underlying issue. And so for me personally, really working on this project, among other things, just because it was fun and interesting, but also really helped formalize and organize how I personally think of ASD. This is opinion, non fact. But so in this particular case, we only have three very specific biological features. I had the synthesis variable, or I guess parameter, I had the clearance, and I had this serotonin-mediated expansion. And even with that, you can get basically any variation on the ENS phenotype, assuming it correlates with a subset of ASD cases. We're making this assumption here. But the idea is, with even only three things, you can basically mix and match them in any way you want. And ASD seems to be actually a very complex genetic condition. Heritability is as high as 0.9%. It's enormous. It's one of the highest heritability factors for behavioral and psychiatric conditions. For example, just by contrast, we all know that schizophrenia and bipolar run in families, but for them, heritability is only about 0.5%. So that was very interesting. And the consensus is that there are at least hundreds of genes involved, but some are estimating up to 1,000 genes involved in this. And now think about this. With hundreds of genes involved, you can have endless permutations to get there, and endless permutations on the phenotype. Even with 3, you can already get basically a full spectrum of supposedly behaviors. You could get so much more in hundreds of them. An interesting thing, it seems to be developing prenatally. This does not seem to be a thing that is happening to one. At the same time, probably some external factors are triggering sensitivities maybe earlier in some people. I don't know. Most of what we know about ASD is based on subtyping people who are around 20, 25 years old. It seems like we only started doing deep dive studies in the aughts. Before then, it was a Wild West. And before 1990s, a lot of ASD cases were classified as schizophrenia. Yes, so really changed in the 1970s, but apparently it was still happening still then. So again, this is how I think about this based on this work. Assume that, for example, each person has a threshold of sensitivity to sound, and another threshold of sensitivity to light, and another threshold of sensitivity to smell, like an individual value of my parameters. And assume you have a population, and all of these sensitivities will fall within some distribution. So, for example, this could be like different people's thresholds of light sensitivity, sound, smell. And then one person will have a combination, will have this threshold of light sensitivity, and this one of sound, and this one of smell. And this one will inherit this feature for light, and this one for sound, and maybe this one for smell. And this person might get this one for maybe high threshold of light sensitivities, and maybe kind of average for sound and smell. And this guy might have really, really low thresholds of sensitivity to all of these things. And maybe by chance, one could get this combination of really high sensitivities to all three. Again, we're just looking at these 3 general features, and I'm sure thresholds of sensitivities are themselves a bunch of genes that are combining and permutating in order to get there. And maybe this way, if you look at this picture that I found, I don't remember where anymore, but I really like it, of the autism spectrum and the different issues or challenges, not just challenges it might have with different aspects. Maybe in somebody with sensory processing challenges might have gotten this combination of these sensitivity thresholds. And now imagine, of course, the combinatorics of this happening not for 3, but for hundreds of traits. And I can just think, well, it must be really hard to form neural pathways when all your sensitivity thresholds are always triggered. Another thing that, and I'm almost done, these days it seems like everyone is on the spectrum. Well, everyone obviously has sensitivities. And also another consideration here is that, as far as I understand, the expanding of this ASD diagnostic umbrella partially was driven by insurance and by the ability to get services and get the support, which is necessary and it helps. It truly does. But so everybody has sensitivities, but it's really the extent to which they compound and the severity and how they affect one's life that actually will determine this. And I found these radar graphs. I do not know this person, but I'm giving her credit because that's where I found them. Of that, okay, so we all have a lot of these things that we are dealing with, how we're interacting with the world. And maybe a neurotypical person would have maybe a little bit of here and a little bit of there. Yes, we all have some sensitivities, but kind of manageable. And this is the going out of this range. So maybe for this one, you would have, oh, I got these characteristics here. And for this one, I got these characteristics. And for this particular feature, for this particular trait, I got these clusters of genes and these clusters of genes and these clusters of genes. And now if you kind of look at the spectrum, so maybe a neurotypical person would just have a little bit of things, and then somebody with ASD maybe would have just many more of, or these things falling in higher sensitivities or something like this, or not compensating for each other, like in the CERT example. And this one, because there is often apparently comorbidity or coexistence of ASD and ADHD, and so somebody might have maybe here and these things and these clusters of genes that are passing along and these clusters of genes that are being passed along. So I found that this visualization really helps me to make sense of this a little bit more, and of thinking about all of these. What if these things are clusters of genes, and they are, because genetics, they are getting passed along and they're getting mixed and matched, and maybe somebody will just have a lot of sensitivities that are passed along to them, but maybe they will compensate for those by, I don't know, hyperfocus on other areas. Opinion, not fact. This is just how I started thinking about this after having worked on this project. Okay, thank you very much. This is the paper. It was published a couple of years ago. This is the experiment that inspired this whole thing. So this is the Margolis paper, the serotonin transporter variant one. I'm not a medical doctor. I'm not a neuroscientist. I'm a PhD scientist. I'm a researcher in mathematical modeling cancer. But it was really interesting. I'm a modeler. I wanted to model an interesting process and learn more about it, and I very much did. Views presented here and my personal views do not necessarily represent the views of my employer or anybody. I do not have financial support on this. This was personal research for fun because I go down rabbit holes. And that will be it. Thank you so much.